Hello everyone. We'll begin with today's session. Um, in the last lecture, we studied about all the upstream processes which takes place for the enzyme production, and those upstream processes include gene identification. So over the last few classes, we studied these all processes like gene identification and then cloning into suitable vectors. We also studied about different vectors and cloning procedures in different host cell, and after cloning it into suitable vectors, we then also studied transformation, that is uh, transforming those vectors into the host cells. And after transformation, the next step is expression of expression of our enzyme. Okay, expression of our enzymes in different expression vectors by different promoters, uh, which are strong promoters, and will give you strong expression of your desired enzyme. And after so this all procedure are considered to be upstream processes. And now when you get optimal expression levels of your enzyme, the downstream processes begin, which includes either the protein can be a intracellular protein, the enzyme that is could be intracellular, that is it could be present in the cytoplasm or different organelles, or you have a extracellular protein, that is it could be secreted in the supernatant where you are growing the culture. So the most of the proteins are intracellular proteins. Very few you are able to make them secretive protein, like that is they get secreted into the supernatant of the culture medium. So since most of the proteins or enzymes are produced inside intracellularly, we need to there are different cell disruption methods. Okay, so this is the first step of the downstream processes, that if the protein is or the enzyme is produced intracellularly, we need to disrupt the cell and get the protein out. So in both the step, if whether it is secreted or whether it is inside the cells, we'll have a crude product, and this crude product will contain many things, like it could contain the proteins which are already present in the cell, cellular proteins. It also could contain protein from the culture medium. If we are providing with different serum in the culture medium, that protein could also be part of this crude product. So the next step is to the next step is to get the solid liquid um, to get the protein extracted from that liquid fraction. Okay, and then we can further concentrate it by using different means of we can concentrate the protein crude extract by using different techniques like evaporation, ultrafiltration, and precipitation. So proteins tend to precipitate. We can precipitate the protein by changing the chemical composition of the solution. And the last step is purification, and we can use different purification techniques. We can use different chromatographic techniques. We'll study about all the chromatographic techniques also in this class. And finally we'll get our enzyme. Okay, so finally we'll get our enzyme, which desired enzyme which we are interested in a overexpressed product we'll get. And then we can do further applications whatever application or we want with that enzyme. Okay, so these are the overall steps which we follow. So first we'll begin with cellular disruption method. So as you can see, there are different types of disruption method: chemical, enzymatic, mechanical, and physical. So let's discuss them. Okay, so there are different cell disruption methods, and this cell disruption methods could be of different types. So we could have physical methods in which you use we have chemical method, and we have mechanical method. So in the physical cell disruption methods, we can use heat. So we can do thermolysis of cells. So we use heat, and we lyse open the cells. So whenever we provide heat to the cell expand and they eventually lyse open, and thus we can get all the intracellular content out. But one drawback of this method is that your protein which you are interested, or the enzyme which you are interested in, it should be a thermostable enzyme. So it should not denature, or um, it should not denature your enzyme, or it should not make your enzyme non-functional. Then you cannot use this method. The next method is by giving osmotic shock. So what is this osmotic shock? So we have different the cell disruption methods for different types of cells. So we have bacterial cells, which are mostly prokaryotic. We have yeast cells, okay, and we have mammalian cells. So so in bacteria and yeast we have a cell wall, whereas mammalian cell and others plant cell also has a cell wall. Okay, so so this osmotic shock will work only for the mammalian cell. Okay, it will not work for the bacterial cells unless and until you digest the bacterial or yeast cell wall. The osmotic shock treatment won't work. So what is this osmotic shock? So you create a different solution. So if we have cells in a solution, you can change the osmosis or change the concentration of the solute here, and this will create a osmotic imbalance, which will lead to osmotic shock, and it will lyse open the cell. The next method is sonication. So this is very well known method. So in sonication we use ultrasonic waves. Okay, we use sound waves which are greater than 20 kilohertz. Okay, so this sonic waves, they disrupt and they open up the cell wall. So these waves create a friction with the water, and they break open the cell wall. So these are all the physical methods. These three are the physical methods which are used. So sonication is used for bacteria, yeast, mammalian, plant cell. Um, so it works with all. In the chemical method, we have different types of chemicals which we can use. That is alkali. So we can use different bases like NaOH or KOH. So what all these bases will do? So the base will dissolve. It will dissolve the lipid membrane. It will dissolve the lipid membrane and it will break open the plasma membrane or the cell wall. So this alkali treatment is one of the chemical method. It's one of the chemical treatment which can be used. The second method which we can in the chemical treatment is we can use different detergents. So again, detergents, the role of detergent and surfactant is to dissolve the lipid membrane or the lipid molecules. So they will either detergents can either completely dissolve the lipid membrane or they can permeabilize. Permeabilization. So permeabilization is nothing but there will be holes in the membrane and whatever proteins you have that will be collected. Whatever this permeabilization will create holes and we will get protein out of the intracellular fraction. And the third which and very important chemical method is enzymatic digestion. So in the enzymatic digestion, what do? We use different enzymes. So like we studied last time also, for cell disruption in yeast we use lysozyme method, right? We use lysozyme method. So lysozyme will break open the cell wall. Lysozyme and other enzymes like glycosidases, peptidases, etc. They will act on the cell wall and they will break open the cell wall or plasma membrane both. Okay, so so yeah, so enzymatic methods are very efficient and you'll very efficient and they are very much suited for cells which have a cell wall, just like yeast and plant cells. So they have very strong cell wall composition. So first we need to do enzymatic lysis with lysozyme glycosidase or peptidase, and then we can do then if the cell wall if the stuffed cell wall is broken up, we can digest the plasma membrane just by doing either osmolysis. We can do osmolysis, give osmotic shock, and the plasma membrane will break open. And the mechanical methods are blenders. We can use blenders just like mixer blenders, and or we can use mortar pestle. Okay, at small scale level we can use mortar pestle, and they will also have, so because of the friction created by these um, uh, mechanical blenders, it will break open the cell wall. Okay, so these are the different so these are the different cell disruption methods which are used in the at industrial level. So at industrial level, they use either bead mill disruptor. So in this bead mill disruptor, as you can see, we have different impellers, and we put in the beads. So these beads will create a friction along with the cell. So we'll add our cell fraction from here, and it will create a friction along with the cell, and it will lyse open the cell, and we can collect our fraction or the cellular homogenate from here. Similarly, in the high pressure homogenizer, we can add a feed or cell culture from here, and as you can see, there is only very small space here, uh, and there is high pressure created at this region. So the cells are sucked in from this small orifice, as you can see here, and as they are sucked in at very high pressure, they lyse open the cell and the homogenized product is collected from here. Now let's go to chromatographic techniques. So once we break open the cell, we'll have a crude fraction. and that crude fraction can be further slightly purified, and then we can do different and the further proper purification uh, at last stage can be done using chromatographic technique. So what is the basic principle of the chromatographic technique? That we have a sample mixture here, and this is our column. So we have a sample mixture and we have a column. Now this column, as you can see, is composed of a matrix. Okay, this column is composed of matrix, and this matrix we also call it as stationary phase. Okay, it's a stationary phase. This blue, it's a stationary phase matrix. So we have packed our column with a matrix, and that matrix will act as a stationary phase, and then we'll have so we have a stationary phase, and then we have a buffer which will add from the sample mixture, that is our sample buffer mixture, and that is our movable phase. Okay, so this buffer phase so chromatographic techniques is composed of stationary phase and a sample mixture. So we'll add a sample mixture and they will mix together. Now the separation will based upon the KD, that is dissociation constant, and that dissociation constant is equal to the concentration of product in the matrix or stationary phase upon concentration of product in buffer. Okay, so this KD will depend upon this KD will tell us how well this two mixture, or how well the mixture of proteins are getting separated from each other. Okay, so if KD value is 1, so that means they won't separate that much. Okay, so for example, let's take this mixture where the KD value is 1. Okay, so they don't separate much. Okay, whereas if the KD value is 9 or 9 or any 9 or a greater KD value, that means they will move faster and there will be a larger separation. Okay, so so this is the basis of chromatographic technique, that uh, they will separate in the phases, in the stationary phase, and this separation will depend upon many factors like the uh, ionic composition of the buffer of the stationary phase, and also how the protein is. Okay, and then we have a chromatogram which is generated. So this is a chromatogram. So we have a signal, and it can be dependent upon time or the volume of the fraction which we are collecting. So these are the different peaks which can be detected by a detector, and these peaks can be eluted depending upon again the KD value. These peaks are eluted depending upon the KD value. So so here A and B are well separated from each other. That means they have a very they have difference in the KD value. Okay, so the difference in A and B is high. That's why they are very nicely separated, and we get a differentiated peaks, very much resolved peaks. But C and D have a KD value which is very close to each other, and that's why they are eluted together. Okay, so these peaks are eluted together. These are the peaks which are not resolved and they are eluted together. So if such things happen, then we have to change our chromatographic technique. Okay, we should use a different chromatographic technique. Now what are the different chromatographic techniques? Okay, so we have different chromatographic techniques. Before that, we'll go into detail about the basis of chromatographic what are different units. Okay, so first we have a reservoir. So this reservoir is nothing but where we can load our samples. Okay, so we can load our samples from this reservoirs, that is this A and B, and this the samples will go from the pump. So what is this pump? This pump is nothing but it will control the flow rate, it will control the pressure, it will control um, all the necessary requirements like pressure, volume, flow rate, etc., which are required for our chromatography to work. Okay, now the third step is the mixer, that is it will mix with a stationary phase. So the buffer, the product will mix with a stationary phase. Okay, and then finally it will get injected into our column. So this column is packed with the stationary phase, as you can see here. And okay, the stationary phase is packed here. uh, in this stationary phase our protein will get resolved, as you can see, and that will be detected by a detector. So if a protein has some fluorescent tags, or uh, it is fluorescent tags, of it's a chromogenic substrate, it can be detected by a detector. And then finally we can collect these peaks. So if we can qualitatively run a sample, and then after at large scale level we can know that okay, the peak is coming at this much volume and or at this much time, so we can collect the fraction using the fraction collector. So these are the different parts of chromatography. So we have different chromatographic techniques, as I mentioned earlier. We have ion exchange chromatography, hydrophobic interaction chromatography, affinity chromatography, and gel filtration chromatography. So what are these chromatographic techniques? We'll go into detail. Depending upon our protein or enzyme of our interest, so if our enzyme has a charge on it, a positive or a negative charge, so we might prefer to use ion exchange chromatography. Or if we know that our protein has hydrophobic patches, or it has a huge hydrophobic region inside, which is situated inside the protein, we could use hydrophobic interaction chromatography. Then we have affinity chromatography, which is very specific and used widely, and it has higher efficiency also. And then finally we have surface area. So depending upon the size of the protein, if it is a tetramer, monomer, dimer, so the size will vary, and you can easily separate the protein depending upon the size. Okay, so we'll go into detail each one of this chromatographic techniques. Okay, so first we'll study ion exchange chromatography. So as I mentioned, what is ion exchange chromatography? So if we have a charge enzyme, like if it has a positive or a negative charge as shown here, so this M is our protein, and so this protein here, uh, for example, this is the mixture of protein, and our protein of interest is M2 minus, that is it has -2 charge on it. Okay, so we can use a positively charged stationary phase. So this is our stationary phase which is positively charged. So when we add the mixture, okay, of this protein, only our interest protein of interest will get bound. Okay, and whereas all the unnecessary extra impurities will get washed out from the stationary phase. Okay, finally we can elute our protein of interest. Okay, as you can see here, we can elute a protein of interest. So the basis is this, that whether your protein is charged or no. So if you have an enzyme and it has a positive or negative charge, you can use ion exchange chromatography. So in ion exchange we have two types, cation exchanger and anion exchanger. So we have two types, cation exchanger and anion exchanger. So what is cation exchanger? The matrix of cation exchanger, that is the stationary phase. The stationary phase will be negatively charged. Okay, and in anion exchanger the stationary phase will be positively charged. Okay, so if our protein is positively charged, we can use a cation exchanger, that is it will bind to a negatively charged stationary phase. Whereas if our protein is negatively charged, we can use a anion exchanger, so that it will bind to the positive stationary phase. Okay, so so this is the detail. So here, as you can see, we have so this is the cation exchanger chromatography. As I mentioned, cation exchanger, our matrix will be negatively charged. Okay, so and here we have a sodium ion bound to it. So the stationary phase buffer will be this is the stationary phase buffer. It will have some positively charged ions which are bound to the stationary phase. So as soon as we add a sample mixture, what will happen? So this is our sample enzyme mixture. So we have a positively charged protein of our interest, and this is our impurities or other proteins which are present. So they will compete with the sodium ion here. Okay, and the sodium ion will be it will be excluded from this cation. It will be removed from this cation exchanger. Okay, and our protein of interest will get bound to this cation exchanger. Okay, to this stationary phase. Now once our protein is bound and the impurities are all eluted out, what we can do, how we can elute our protein of interest? By adding excess of Na plus ion. So we can increase the Na plus ion concentration, and then what will happen, this Na plus ion will compete with this cation exchanger and it will elute our protein of interest. So this is the basis of ion exchange chromatography. Similarly, in an anion exchange, we will have a positively charged stationary phase, and here we will use a different buffer. Okay, we will use a chloride ion buffer. So this chloride ion will get bound to the stationary phase, and when we will add the protein of our when we will add the crude protein, so this crude protein, if we want to separate this red protein from here, what it will compete with this chloride ion, right? And the chloride ion will be excluded, and the protein of interest will get bound to the stationary phase. And so the steps in the ion exchange chromatography are very simple. So first we'll have a stationary phase packed. Okay, so we'll make a stationary phase, that is we'll pack the column. So if this is a column, we'll pack it with the stationary phase, that is it will contain the beads. It will contain beads which could either be cation exchanger or anion exchanger. Okay, after packing the stationary phase, the next step is to prepare the mobile phase. Okay, to prepare the mobile phase, what is this mobile phase? We'll add this buffer. Either it could be Na plus buffer or it could be Cl minus buffer. Okay, depending upon the type of chromatography, it could be either Na plus or Cl minus. Okay, the next step is adding the buffer. It could be either Na plus or Cl minus. Okay, the mobile phase. The third step is then we'll add sample. Okay, we'll add our crude protein. Okay, so we'll add a protein of interest, and it could be either this or this protein, whichever protein we want to purify. And then the last and final step is the elution. So in elution, what we'll do, we'll add same buffer, but we'll increase the concentration of these ions, we'll increase the concentration of the buffer. So if we are using NaOH, um, so if we are using this Na plus buffer, any Na plus buffer at 1 molar, we can just increase the concentration to 10 molar. Okay, this is just an example. We can increase the concentration, so there will be more Na plus ion, and these Na plus ion then further will compete and it will elute our analyte, okay, or our enzyme of our interest. Okay, and thus we'll get so the finest will get the elution, so in the elution volume we'll get the enzyme. So this is how we do ion exchange chromatography. Then we have different types of we could either use a strong cation exchanger, that is sulfonic acid group which is present in this strong, and these are the different examples. SP cepharos, this SP stands for sulfonic acid, SP cephadic, CM cellulose, and CM cepharos. Or we can use a weak cation, which is the weak cation is carboxylic acid. So these are the examples of the weak cations, and there are different pH range given. So you should also consider that depending upon the pi or the pH range. So if the protein is positively charged or negatively charged, depending upon the isoelectric point, this pH range are used for different cation exchanger chromatography. Similarly, we have strong anion, that is quaternary amine, so they will have very strong interactions. Okay, and the weak anion exchangers are primary amines, secondary amine, tertiary amine. Okay, like DAE cepharos, this is very widely used, DAE cepharos. Next, we move on to hydrophobic interaction chromatography. So what is this hydrophobic interaction chromatography? So the protein is folded in such a way that all the hydrophobic patches or hydrophobic interactions are inside the core of a protein, whereas all the polar interactions are present on the outside of the protein. Okay, all the hydrophobic interactions are inside the core, and the polar groups or polar residues are present outside the proteins. Okay, so this is how the protein folds. So this is how the protein is folded. Okay, so the principle of hydrophobic interaction is uh, when we add a ionic solution like NaCl or NH4Cl. Okay, what will happen? So it will increase the ionic strength. And that will increase the solubility. Okay, once as we increase the salt concentration, the solubility will increase. Okay, here till this phase the solubility has increased, it has gone up. Okay, now this process of where the solubility is increased, okay, solubility is increased as we add, okay, as we add salt, as we increase the salt concentration, this process is known as salting in. Okay, at some point what will happen, so this is the point where how much ever salt you add, so this is the point where is how much ever salt you add, there won't be any further increase in the solubility. Okay, there won't be any further increase in the solubility. So this concentration is known as CMC, that is critical micellar concentration. Okay, here there will be a micelle formation. Okay, and if further increase in the salt, there won't be any increase in the solubility. And as you keep on increasing the salt concentration, what will happen? The solubility is decreasing. So what is happening now? All the as you increase the salt concentration here, so this is the all the water molecules will move out of the hydration shell, okay, and the protein will get precipitated, okay, and this process is known as salting out, okay. The protein will get precipitated because all the hydration shell all the hydrogen all the water molecules are removed from the protein, and this will disrupt the protein structure, okay. This will disrupt the protein 3D conformation, and the protein will get precipitated out, okay. And after getting protein precipitated out, it will expose the hydrophobic residues. It will expose the hydrophobic residues, okay. So once the hydrophobic residues are exposed, we can do the hydrophobic interaction chromatography. So as you can see here, so we have a matrix. Again, we have a stationary phase here, and to this stationary phase we have a receptor, and this is a hydrophobic receptor, okay. So this receptor, as you can see here, it's a hydrophobic receptor on the stationary phase. So we have a hydrophobic receptors present, and what will happen as we add high salt, um, there will be salting out, okay. Once we had higher concentration of salt, if we add uh, salt concentration at this level, what will happen? Salting out will happen, okay. And these are the hydrophobic residues. I'll change the color. So these are the hydrophobic residues. This dark red color are the hydrophobic residues. So when we had high salt, what will happen? These hydrophobic residues will get exposed outside, okay. They will come out of the protein core. So these hydrophobic residues now were inside, they will come out of the protein core, okay. And they will react with the hydrophobic receptors, okay, which are present, and they will get bound to this stationary phase, okay. They will get bound to this stationary phase. Now after these protein molecules are bound to the stationary phase, what we can do, we'll add a low salt. We'll decrease the salt concentration. We can decrease the salt concentration. So from here we can decrease the salt concentration to here. Now here the solubility is the highest, right? The solubility is the highest. So all the hydrophobic residues now will get, again it will get inside, and the solubility will increase, okay. And it will no longer be bound to the protein. So this is how we elute the protein. So this is how we use this hydrophobic interaction to elute out the protein. So first is we prepare the matrix, then we add our sample, we increase the salt concentration. So these are the steps that we first prepare stationary phase, then we add the sample, that is our crude protein, then we increase the salt concentration, then if we increase the salt concentration, what will happen? It will expose the hydrophobic patches. And then it will get the protein or the enzyme of our interest will get bound to the stationary phase. And once it has bound, we can do the washing to remove the We can do the washing to remove the unbound protein, and the final step is elution. So how do we elute it? We'll just lower the salt concentration. Okay, then our protein of interest will get eluted. So this is how we do hydrophobic interaction chromatography. Next is gel filtration chromatography. So when do we do gel filtration chromatography? When we have different sizes of protein of our interest. So if we have a mixture of protein where the protein is of different size, okay, and we want to elute out a very small or very huge protein, we use a gel filtration chromatography. So suppose we want to elute out this very small protein, very small size protein, okay, as compared, so the size is very small. So what will happen, so we have a beads, the stationary phase, as you can see, it's made up of beads, okay, beads, and these beads have a sieve like structure, okay, these beads have sieve like structure, they are porous in nature, okay, and these beads have this sieve like structure, and the stationary phase is completely filled inside inside this porous beads, okay. So these are the beads which I am talking about, and these beads, what will happen, the smaller protein, okay, so the smaller protein here which we want to separate, the smaller protein, they will go into the sieves like structure, okay, and they will come out, they will take a lot of time to come out from these beads, okay, they will take lot of time come out of these beads, whereas when the protein is of huge size, if the protein is of huge size, they will not go inside the beads small pores, and they will just slide off of these beads, they will just slide off, okay, from the side of the beads, they will not go inside, and they will elute faster. So this is the basic principle of gel filtration, that the larger molecular size, that is the larger proteins, okay, the larger proteins will get eluted faster because they will just slide off these beads, okay, they will just slide off from the beads, whereas the smaller proteins, okay, smaller proteins, what will happen, they will go through these sieves, okay, and they will travel a lot in these beads, and they will take a lot of time to elute, okay. So as you can see here, the smaller green beads are still traveling inside these porous beads, and they will take a lot of time to come out, so the larger molecular weight will get eluted out faster, okay, as compared to the average molecular weight, and the smaller molecular weight will elute the last, okay. So this is what the chromatogram will look like, the larger molecular weight will elute out faster, so this is a time or volume, so this will elute out faster, this is a medium size, and the small protein will take a lot of time, and they will elute very late. So this is how we can separate proteins based upon the size, okay. So this is the basis of gel filtration chromatography. And then finally we have affinity chromatography. So affinity chromatography is one of the it's very specific chromatography, because it is highly selective to the protein, the enzyme which you want to separate. So what does it so we have a receptor and we have a ligand. So this receptor has a very high affinity for ligand, and we are using this principle in the affinity chromatography, where in a stationary phase, so if this is a stationary phase, it is bound to a receptor, okay, it is bound to the receptor, and this receptor this receptor has a very high affinity, it has a very high affinity for the enzyme, or the protein of our interest which we want to separate. Okay, so what will happen? They will interact with each other by different interactions like salt bridges, hydrogen bonding, van der Waals, just like enzyme substrate interaction. Okay, just like enzyme substrate interaction, they will also interact with each other. Okay, and these are very high, these interactions will be very specific, that is they will be of higher affinity. Okay, and that's why the protein, you can separate n number of proteins. So if your protein cannot be separated by other techniques like ion exchange or size exclusion chromatography, you can use a affinity chromatography to separate out the proteins. For example, if the protein is of if the protein doesn't have any if it is not either small or large proteins, so you cannot use a gel filtration, or if it doesn't have any charge on it, and ion exchanger chromatographies cannot be used. So you can use affinity chromatography, and it has various advantages also. So so this is how the affinity chromatography works. So this, we have a matrix, and we have a ligand which is immobilized, or we can say a receptor or a substrate which is very highly specific to the enzyme which we want to separate by using affinity chromatography. So again, the first step in the affinity chromatography is the stationary phase preparation, and the stationary phase here is the matrix which contains the immobilized ligand. Okay, so we have this immobilized ligand. Okay, and the free enzyme will come, and it will have a affinity for this um, ligand. So the second step is to add sample which will contain our enzyme specific to this ligand. Okay, and how do we elute this? The elution is very simple. Either you can use different you can change the ionic concentration so that these interactions will become weak, and there will be separation. So you can use different ionic concentration or change the pH of the elution buffer. So elution is also very simple. You can change the ionic concentration or pH, and and then and then you can collect the purified enzyme. Okay, or the next step is dialysis. So dialysis is also very simple. In dialysis, the interactions get denatured by different concentration of ionic salts. So you can separate this enzyme substrate interaction or enzyme ligand interaction by doing dialysis. Now this receptor ligand follows this. It will again depend upon the KD. So this is a receptor plus ligand. It will give a receptor ligand complex, right? So this is receptor, and the dissociation constant will be equal to concentration of receptor ligand upon complex. Okay, so you can change this KD by changing salt concentration, ionic strength, or also pH. Okay, so this you can change this dissociation constant based upon then, and you can get your purified enzyme at the end for to do elution. So these are the different receptors, and they have affinity for proteins. So we have 5-AMP, which has affinity, 5-AMP receptor which has affinity for this enzyme NAD plus dehydrogenase. uh, then we have this very popular avidin and biotin containing enzyme. Poly A will get bind to poly U mRNA. This is um, RNA hybridization, if you can say. Um, Similarly, lecithin will get bind to glycoprotein. Heparin can be heparin can be so if we have a DNA binding site which has a specificity for heparin, so we can separate the heparin also. So what are the advantages of this affinity chromatography? So we have different advantages. First and most important is specificity. We can specifically purify the enzyme of our interest, like NAD plus dependent dehydrogenase or any biotin containing enzymes or immunoglobulins. Okay, so specificity is very high in this type of chromatography. Also, the yield which we get, the purity yield is also very high. And the third is that it's highly reproducible. Once you have the chromatography overall structure ready, it is highly reproducible also. And and it's easy to perform. It's easy to perform and easily eluted. So the overall procedure is also simple. Okay, and there are two types of affinity chromatography. We have a bioaffinity and we have a pseudo affinity chromatography. So we have a bioaffinity and we have a pseudo affinity. So in the bioaffinity, we use biological molecules like enzyme substrate, and immunoglobulins IgG and antigen antibody interactions. Whereas in pseudo affinity we use metal ions and different dyes we use, okay, to separate. So how can we generate antibody for, let's take this as example. How can we generate antibody for to make antibody to separate out some antigen? Okay, so if we want to have a stationary phase, and we want to have um, if we want to make antibodies, which will bind to the stationary phase and it can separate out a protein of interest. So the protein crude mixture will get bound and it will get separated, so the purified antigen will get separated. Okay, so how do we make this antibodies? It is very simple. We can do a antigen presentation in the any animal model. That is, you can put the antigen inside the model organism. It could be either rat, mice, rabbit, and then we can raise the antibodies. We can generate the antibodies and we can collect the blood sample and purify the antibodies present in the blood. Okay, so it is very simple, and then you can immobilize those antibodies onto the matrix by using some spacer or a receptor. You can immobilize them and then use it for affinity chromatography. Okay, with this we move on to the assignment question. Okay. The organic solvent is used to disrupt the plasma membrane is organic solvent is used to disrupt the plasma membrane is acetone. So acetone is the organic solvent. It will disrupt, it will dissolve, the lipid bilayer. Okay, and it will dissolve the lipid bilayer and it will disrupt the plasma membrane. So answer is acetone. Osmotic pressure generated in the solution is by difference in the solute concentration, salting out, salting in, same solute concentration. So we can have difference in solute concentration. For example, we have cell, okay, and the cell has some osmolarity maintained at 7.2 pH, okay, and this osmolarity is maintained by the cellular composition. So if we add, so cells will grow well in PBS, which has same pH around 7.2 or 7.6, okay, but now we add a different, suppose we add water in this, okay, if we decrease, if there is a difference in the solute concentration, so now this solute concentration here is same with PBS, but if we change the solute concentration, it will lead to osmotic pressure. If we increase the water concentration here, what will happen? Here there will be, inside the cell there will be less water concentration, so all the water molecules will move inside the cell, okay. So we have water molecules here, inside we have a aqueous environment in the cell. So if we increase the water concentration outside, what will happen? This water will move inside, okay, because of the difference in the concentration until it reaches equilibrium. Once the water has moved inside, what will happen? The cell will increase in size and finally it will burst open, okay, the cell will burst open, okay. So this is how osmolysis occur. Osmolysis. So difference in solute concentration can lead to osmotic pressure change. Osmotic shock is used to remove this periplasmic. It is used to remove periplasmic substances. Very simple. The chromatographic technique that depends on highly specific interaction between pairs of biological materials such as enzyme substrate. This is just now we studied. Highly specific interaction is affinity chromatography. Okay, they will have affinity just like enzyme substrate. Okay. Dash allows chaperones to recognize a wide variety of misfolded structures. So ATP hydrolysis is required. This energy from the ATP hydrolysis allows the chaperones to recognize different misfolded protein structures. So ATP hydrolysis is the answer. The interaction that contributes most to protein folding are covalent, hydrophobic, ionic, van der Waals. So all the interactions are important, but the most important interaction is the hydrophobic interaction. Okay, so hydrophobic interactions are the most important in order to get proper protein folding. Chimeric humanized monoclonal antibodies are generated using hybridoma technology. Okay, the correct answer is hybridoma technology. rDNA technology is used for DNA fingerprinting, not for antibodies generation. Southern blotting is also DNA hybridization. Immunization is Immunization is different, where you expose antibodies and antigen in order like to generate vaccines and to make vaccines, so that is completely different. Match the following. So we have a receptor in this section and we have an affinity towards proteins. So as we studied in the affinity chromatography, we can use this for affinity chromatography. So avidin has higher interaction with biotin. Poly A will hybridize with poly U mRNA. So this is RNA hybridization. Lecithin will hybridize with glycoprotein. This is interaction found in the cells also. Heparin identifies the DNA binding site. So A3. So the correct answer is 3. Dash is not highly specific biological interaction to be used in affinity chromatography. So in affinity chromatography we do use enzyme substrate because it is very specific. um, um, Antigen antibody is again very specific. Receptor ligand is also very specific. Cations anions is not that specific because Na plus will also react to the cation Cl minus, as well as NH4Cl can also interact with Cl minus, so it won't be that specific. Any positively charged cation will react with negatively charged anion, so it's not specific. So that's why ion exchange chromatography is less specific than affinity chromatography. Dash tag is a 6 amino acid residue peptide which can be immobilized by transition metal ions. So the correct answer is his tag. So in his tag we have 6 histidine residue, and they react with nickel. Okay, so this is a pseudo affinity metal affinity chromatography. They react with the nickel ions. Okay, and these technique is very highly used in many mol bio. So we have a protein with a his tag which has a 6 histine residues on the N terminal or C terminal, and then they use affinity chromatography where the stationary phase has a nickel NTA column. Nickel NTA column in the stationary phase is packed with this nickel NTA beads, and the protein will get bound to this. The 6 histidines will get bound to this. Two monomeric protein migrated identically in SDS page but got separated in size exclusion chromatography. The reason would be, so here they are saying two monomeric protein migrated identical, that is on the SDS page. So basically monomeric proteins are migrated identically on the SDS page. So SDS page is a denaturing page. Okay, what it will do, if we have a trimer like this, tetramer like this, or trimer like this, what it will do, it will denature and we'll get monomers like this. Okay, we'll get monomers, and these monomers, as you can see, they are of same size. Since they are of same size, when we run on page, what will happen, we'll get a same size bands. We'll get a same band, we'll get only one band. If we run this, if we run a trimer and if we run a tetramer, we'll get a same size band. Okay, equal same size band, they will run in the similar length. Why? Because SDS is a denaturing agent. It will get migrated identically on the SDS page but got separated in the size exclusion chromatography. The reason would be, so in size exclusion, it has, so size exclusion again, it will based upon size. So also, even the if the size can increase if the protein is in the form of a tetramer or a trimer. Okay, the size can increase. So the option is D, that one protein is monomer whereas the other is a tetramer. So in the SDS page they are getting denature and we are getting identical band, but in the size exclusion chromatography there is no denaturation as such unless we do it, but there is no denaturation and the size will remain intact, and these size will get separated based upon um, the molecular weight. So one protein has to be a monomer and another a tetramer. A globular protein of molecular weight 15 kilodalton exists as a mixture of monomers and dimers in a solution. The most appropriate technique for the separation of two protein, again uh, what I explained before, that whenever there is a difference in size we go for gel filtration chromatography, right? So thin layer, paper and ion exchange chromatography, they all work on the charge of protein, charge on protein, whether the protein has any charge. So here they have not mentioned anything about the charge, whatsoever the charge is, but there is a difference in the molecular weight and the size. Again, it's a mixture of monomers and one is a dimer. So obviously the size is different. One is 50 kilodalton and other is 100 kilodalton. So this protein 100 kilodalton will get eluted first and smaller protein will get eluted last. Okay, so gel filtration is the best to separate the two forms of protein. The procedure in which cell lysis occur because of shock waves which are formed due to collapses occurring in the buffered solution containing cells is referred to as. So shock waves are created in ultrasonication. Okay, so ultrasonication uses shock waves more than 20 kilohertz, right? So ultrasonication. Alkali treatment uses NaOH or KOH. Osmotic shock, again we uses difference in the solute concentration or pH. Detergent, again we use SDS. So they will lyse open, they'll dissolve the cell wall and lyse open the cell. Digestive enzymes, a digestive enzyme, salivary amylase, in the saliva begins the digestion of carbohydrates, protein, fats, nucleic acid. So salivary amylase, as you can see the name, it has amylase. So amylose structure is found in starch, okay, which is a carbohydrate. And salivary amylase will digest carbohydrates, okay, not protein, fats or nucleic acid. In electrophoresis, if the buffer pH is above isoelectric point of the protein will migrate towards cathode, migrate towards anode, not migrate at all, migrate partly to anode and partly to cathode. So we should remember that if a protein above the isoelectric point, it is negatively charged, and below it is positively charged. So here the pH is above the isoelectric point, so we must know this protein will be negatively charged. Okay, now cathode is negatively charged, cathode is a negatively charged, anode is a positively charged in the battery, so it will migrate towards anode. Okay, so negatively charged protein will migrate towards anode, very simple. Always remember, anode is positively charged, anion is negatively charged molecule, anion. Okay, anions move towards anode, cations move towards cathode. Okay. Any questions? Okay, then we'll stop for today if no more. If there are no questions.